Well, good morning again. We are here again, and we're going to talk about today from Haplaxius crudus as insect vector of lethal yellowing in Cuba. As all you know, lethal yellowing is the most devastating disease affecting several palm species in the Americas, and its occurrence is mainly associated with the presence of phytoplasmas of the 16SR4 and is also known the insect Haplaxius crudus as the mainly insect that was confirmed as vector in the Florida region. In Cuba, previous work informed the presence of phytoplasmas of the group 4, and more recently, studies performed during the Tropicsafe project show that several groups of phytoplasmas were detected in the sampled coconut palms in several locations of the country, with a high prevalence of the group 4 in these samples. Also, it was consistently found Haplaxius crudus mainly in the coconut farms in the western region of the country. However, despite the frequent presence of this insect, a low population level was also confirmed, and these antecedents are because we trace the objective to investigate the role of Haplaxius crudus as vector of phytoplasmas on coconut ecosystem in the western region of Cuba. What we do for that? Well, we obtain the plant material using healthy seedlings growing under greenhouse conditions. The seedlings were confirmed healthy using this nested PCR system well established in our lab, and the insects were individuals of Haplaxius crudus collected on the field, mainly on the western region. This was mainly by two reasons: the COVID situation in our country and the presence of the insect in this area, mainly Alquizar and Hawaii Grande. What did we do? Well, we performed 3 asides, 1, 2, 3, 3 thinly of Prechardia and Cocos nucifera in the last 2 asides were used, and approximately 50 insects per plant were used. These systems were caged, were put on the greenhouse condition, and after that, insects were individually recovered per plant and per aside, and the plants were maintained under greenhouse conditions to periodic monitoring for symptoms appearance. The DNA extraction were performed using the CETAT method, start point of plant, midribs of the leaf, and for the insect, a pool of 15 insects were used. For the PCR detection, we use a nested PCR. As you can see, the applicants obtained a wet purified using this kit and were direct sequencing, and the sequence were used for analysis of phylogenetic and analysis of the sequence using BLAST, MEGA, and I5 classifier. As you know, all the routine of identification of new isolate found of phytoplasmas. Well, after several months post-inoculation, on the first 2 asides, the exposed plant didn't show symptoms, as you can see here. However, one of 3 plants used in the 3rd aside showed mainly foliar yellowing in this plant, as you can see in the picture. This was confirmed using the PCR analysis as the plants of the first and 2 asides, and also the insect used in these asides were negative for the detection, and in the case of the 3rd aside, only the plants that showed symptoms and the used insect in this aside were positive for the detection of phytoplasmas. These 2 sequences were purified sequence and used in the analysis next. The two sequences show 100% of similarity to each other, and also the blast analysis showed the highest percent of identity with strain of LY phytoplasma previously informed, as you can see in the table. Also, the similarity coefficient calculated on the I5 classifier support the belonged or isolate to the subgroup A of the group 4 and its relation to Candidatus phytoplasma palmae reference strain, and this means that this strain belonged to this reference or are related with this strain based on the 16S sequence. Also, the RFLP pattern was quite similar to the subgroup A except for these two enzymes, which originate slightly different bands, as you can see here, pointed with the yellow and the red arrow on this picture. Also, the phylogenetic analysis located all two isolates on the clade of the 16SR4 subgroup A, showing the relation within it. This result confirmed for the first time the Haplaxius crudus as a vector of LY phytoplasmas in the coconut ecosystem in Cuba, and also the phytoplasma involved in the transmission trials was strongly related to the subgroup 4A. Well, thank you for your attention.
My name is Camilo. My partner is Maritza. We work at the Research Institute of Tropical Fruit Crops from Cuba, and today we're going to talk about HLV symptomatology and methods to contain the disease used in Cuba.